Hello, everyone. Um, Welcome to my presentation for Node 2 2021. This is going to be about building a graph of basically gene and variant links between two species. So without further ado, I'll go into it. So I'm doing some work under a product called GeneWeaver, and GeneWeaver is actually wider than just this graph, but there's a little picture that one of the researchers drew on the whiteboard with my colleague just before I started at the organization, which was kind of the thing they wanted to see, what they really wanted. So what they actually had was a kind of a tabular database, basically a more traditional type of database. And what they really wanted was to be able to see the relationships between them, between genetic entities, and that's what I'm going to talk about today. Probably should say this is a collaboration between Jackson Laboratory and Baylor University and over in Texas. So who the hell am I? So I'm going to talk about some genetic stuff, but I don't actually know very much about it. I'm more of a software person, mostly working with Java in the past. I had around 10 years, I worked with finite element analysis, including computational fluid dynamics. And then after that, I worked at a physics experiment called a synchrotron, where we looked at, we had detectors and we looked at different patterns produced by light when you pass it through samples. Then for a couple of years, I worked in geology, so stratigraphy using machine learning. And last year, I moved over to this project. So this is working for Jackson Laboratory, and that's genetics, basically. The project is, JAX itself does lots of different things, but this project is to do with genetics. And I've also doing a few other projects with ML, but Graph has certainly been a major part of what I've done working for them. What I'm going to show you today is open source, so it's available on Bitbucket. We're partially funded publicly and funded by also philanthropists in what we do, so a lot of our code is available open source. You can get the binary artifacts on Maven Central if you wish. So they're not actually checked for intellectual property, but one day we would like to do that, and I plan to work with the science working group at eclipse.org, because I've worked them in the past and I know that they're really great for checking IP. So that's kind of where I'm coming from. The organization I work for is called Jackson Laboratory, and I wanted to just fill you a bit in on what that is, and the kind of perspective I have on this graph stuff. So JAX itself is a non-profit biomedical research institution, and it discovers precise genomic solutions for disease and empowers a global biomedical community in the shared quest to improve human health. It does basic research, and it also does education. And as I said, it's funded both by research grant, which is that'll be merit-based, based on scientists applying for to look at different areas, and it's also funded by philanthropy. People donate to our facility in order to help find cures for diseases and things like that. So I'll talk about in a bit just about some of those aspects. JAX itself is mostly situated in the US, Maine, Connecticut, California, and there's an office in China. I'm actually working in, as you can probably tell from my accent, I'm working in the UK at the moment, but I'm planning to move to Maine in a week and a half. So lots of changes for me. So I wanted to drill into a couple of the areas that JAX works within, and why we're interested in this graph. So around 50 of our researchers are looking at cancer, and that's both the kind of reasons why cancers develop and possible treatments and things. So that's one of the key areas, and I just want to drill into that to say that's just give you an example of what it is that we're actually doing with our research. Another example would be looking at Alzheimer's. So around 40 researchers are working in wet labs looking at the causes and possible resolutions of Alzheimer's disease. I mean, another example would be the roots of addiction. So people scientists are also studying that, and many of them at JAX are looking at why some people get addicted to some things, and that too has interesting genetic backgrounds. So the other part of what we do is education. So it's not just pure research; it's also people attend the site to do courses. And as you can probably tell from my background, I'm doing some at the moment to try and get a bit more trained in this area. So the graph stuff that I want to talk about was published in a paper in Nature by some of my colleagues, and I'm working on their team now. So Tim Reynolds, Jason Bouvier, Alyssa Chesler, and Eric Baker are all sort of working with me. And that that paper handily drew a graph. So if you see in the bottom left-hand side there, you can see a mouse and a human, and there's some links between them. And it could be another species, but we're focused primarily on mice at JAX, and and how they link to humans. But the actual wider graph could be any species and human. And so that if you look at disease in that species, you can infer things in the human, and vice versa. So if you have a known issue in a human, you can look at where the genetics of that in the kind of animal model for that. So a lot of they pointed out in the paper that a lot of this data actually exists and could be in a graph form and allow researchers to find links. But it's in lots of different file formats, as they say in the top half of that on the diagram on the left. There's all the different file formats that the data is actually in. So the rest of this talk is going to be how do we get that data into Neo4j. Okay, so first of all, what is that data? So it's in the case of human beings, there's around 100 gigabytes of files in different formats, mostly zipped up text formats. So obviously, coming from physics, I'm a bit more used to binary formats for data, but no, I have to actually parse the data and create the graph that way from FTP sites with lots of lots of data on them that kind of been encoded. I'm going to show you some of the ways that data is made available to researchers. It comes from a data set called Ensemble. There's one called GTEx. Jackson Laboratory itself produces some of the data, especially around mice, and there's a project called Oncode. And in fact, there's several other projects that provide data for this. And the data is very heterogeneous. I'll talk about the scale in a minute, but the scale of this data is probably the scale of all scientific knowledge on genetics of humans is much smaller than the scale of a network on something on a big social media site. So that gives you some interest there in terms of how big this problem is. It's actually an interesting perspective. But the input files themselves are lots of kind of zipped up text files, as I said, and they are usually separated. They're table files separated with a character such as a comma or a space or a tab. But also within that, they may have data which is in a list form separated by a semicolon or something like that. So you've got hundreds of gigabytes of data that you have to run through and somehow build a graph from that. So that's kind of describes the problem a bit, I guess. So taking a step back from the problem, why would we look at graph, apart from the fact that the researchers draw graphs when they talk about this data? So obviously, we need to be able to ingest data of this size reasonably quickly. And that's fine. All of the technologies that you might compare to graph are going to be fine for that. But the other thing we want to do is once we have this data, we want to really talk to it in a graph format. So that might be GraphQL, it could be Cypher, but we want to have some ability where we're not continually transforming, nodes and edges into some pseudocode that then is transformed back into nodes and edges. And this is a key point. So graph databases support that kind of stuff, right? And we want something that's equally efficient for non-tree shaped data. So NoSQL databases are very efficient if your data is a tree, and our data kind of is a tree, but it's a tree with roots, right? So it's got a tree on both sides of the trunk, if you like, and actually there's a few cross linkages which I'll talk about. So it's not that, it's not great for that. So again, graph doesn't care, right? It's just nodes and edges. It doesn't matter about the shape. The next point is that we don't really want to have to join tables at different points. We just want to be able to traverse our connections. And the final point is that we want to ingest new data efficiently. So that might mean quite complex nodes and edges interacting with existing data. And this is where, again, graph is highly interesting to us, right? So I guess at the end of that, and probably a few other discussions, we were thinking we need some kind of graph database to solve this problem. So then the question is, why Neo4j? And when you parse all this data in, it comes out as around roughly 1 billion nodes and roughly 10 billion relationships. So Neo4j will easily scale to that. Even the community edition scales that far. So the other thing about Neo4j is it has a REST API, so that allows us to send Cypher into an endpoint and get back a table. So that means you can have a nice little shell script or something like that with a curl command that a researcher can push in a kind of prototype graph. And very often they have input files which are may have hundreds of thousands of gene variants in, and they want to get a table back for that. So that's the kind of thing that the REST API in Neo4j gives you. It's very, very cool, and it's perfect for our use case. The speed's fine. Some of our query, I guess a typical query for us might take 20 to 30 minutes. So, but some of them obviously are small. It depends on how much data you push into the graph that you want to get out, obviously, and also what the indices are that you've created on that database versus the query that the user's making. And I think another key thing about Neo4j, is the previous speaker Antonio mentioned, was that this APOC functionality is very useful. And for us, we want to analyze tissues and things later on with our graph, and APOCs will give us an ability to do that. We also might be interested in some of the machine learning stuff that Neo4j has. Prototypes previous to the one I'm talking about, we had one based around an SQL database, and what that did is it read all of this data into memory, because you're not really doing biology properly unless you read everything into memory and use a supercomputer. But that then is written out to the tables, and then that gives you pretty much most of the functionality of the graph by having queries that you can call on it. The thing about that prototype was reproducibility. So we want to take these data sources, change all the time. People are always experimenting and producing new input data. And we reckon that every 4 months we need to rebuild this graph entirely. We basically throw away the old one and download all the new data and then rebuild the graph. Because science is moving forward that quickly. That's roughly the cadence that we think. In fact, since I built the last version of this at Christmas, another version of some of the key files has been released. So that that kind of ties with that idea. So any graph that we build must be kind of automated. It must be, you must run a script to download. You run a script to ingest that data, and you end up with a very reproducible graph, which is of known provenance. You know where all the data came from that made that graph. Another prototype that we made actually at JAX with my colleague Jake Emerson was based on Python, and it streamed the data from these input files and used transactions. Now, that was a good basis for what we later did, but it was rather slow. So I wanted to go on to how we managed to speed up building this graph in a reproducible way. So I played around with Jake's code last year, and I swapped it over to Pi to Neo, and using the OGM. So a lot of these input files will have, let's say you have a gene in the file, and then you have a few transcripts, and they link back to the gene somehow. So the file has got multiple entities. It's got nodes and relationships within a flat table. So we want to kind of stream those objects and then create the graph. And if we were to use transactions, it was around 8 and 9 milliseconds a node. And so the time, and that was based on our benchmark of 1.7 million nodes. So the time for the full graph being built would be about 100 days. So we can do 3 a year. So that's too slow, right? So I moved it over to Java and did the same thing, basically, just a single thread, and parse the files in a, using a stream. And that, just by doing that, it brought it down to 57 days. But obviously that's too slow. So then I used, rather than one commit per object, I committed based on transactions, and that halved it again. And then used multiple threads. And then used a chunked read, which it has some of these files are kind of semi-ordered in that a gene is always followed by the transcripts that belong to that gene. So you can actually optimize around that and read a whole gene chunk, and that actually speeds up the building of the graph. And it kind of got it to 2 or 3 days. So that wouldn't be too bad. But another colleague is using Neo4j on another project, and they said, well, you know about bulk import? And I went, no, I don't. And so they explained to me that there was this bulk import feature, and that rather embarrassingly, I'd spent time optimizing something which there was already a way of doing. But actually, it turns out that some of our fast transactions will be needed later for updating the graph, so few. But we moved over to bulk import, and that just takes a single thread and parses these input files, and then writes lots of output files. I've got a slide on that, and that takes it down to around half a day to a day. So the kind of thing where you can run it, and the next day you'll have a graph. So all this stuff obviously relies on streaming. We don't really want to hold the data in memory like one of the earlier prototypes. We want to be able to stream the data. And so there are some packages for doing that, but I wanted precisely the most efficient stream for this use case to get those times down. So I wrote a IO package for some existing file formats, which do have readers in Java, but for whatever reason, we're not optimized for doing this kind of thing. So all of the stuff I'm going to show you is basically based on streaming from those files using Java streams, and then you're kind of mapping between those. So next slide. So what does the bulk import look like? So it's a command line interface. So you log into where you want to build it. You have a Neo4j available, and then you download the data and you run the command line interface to build the files. It's not quite as straightforward as all of the data being transformed directly, because it's too big to fit in memory, as I said, and some, unless you use a big memory machine, but a lot of those things can be cached. So there's actually a database that gets brought up as you're writing these files and caches them for later objects so that you can create the correct links. And then I don't really want to go into detail about the bulk import feature of Neo4j, because there's a fantastic tutorial online. So I would encourage anyone who's thinking of using bulk import to go and have a look at that. And obviously our use case has stretched it a little bit in that it's continually rebuilding quite large data. But again, it's much smaller than many social networks or things like that. So it's perfectly doable. One thing to note when we were building these files that Neo4j uses for bulk import is that for us, at least for our use case, Neo4j4 was quite a bit faster. So the Neo4j admin command was able to import to Neo4j4 quite a bit faster than 3. So it's worth noting. So a bit more on bulk import is streaming these objects, as I said, and then it's mapping them. But it's not quite as simple as that, because multiple objects come out, of course, and then those objects have connections, which you need to write the connections files for those objects. So the stream of objects coming from a single file is then connected using a flat map. So let's, if you imagine a gene comes out of this file, and that will be a gene object in Java. That gene object will then have connections, which will be multiple objects, right? There'll be multiple relationships to other objects. So the connector then goes and finds out that, and you end up with multiple. So it does a flat map. So it goes from a stream of genes to a stream of genes plus the connections. And in fact, the original stream is heterogeneous. So it might see a transcript and map, and then map back to the gene. It might need to go the other way. But at the end of this, all of these entities are coming out in a stream, and they then are saved to their individual bulk files. As I already mentioned, in some cases, a database is written that there's different tables. Some of them are around a few gigabytes, and some are only a few megabytes. So that's all very well. So we've managed to take all these input files and then build them into the files that Neo4j can slurp up. That's great. But how do we actually deploy that? I mean, what does it look like for users to use it? And that was a bit of a question for us, because we have a bit of an unusual use case. And that is that we have a low number of users doing a large number of queries. So one user may run 100,000, may import a CSV file looking at 100,000 variants, for instance, and that might just be one interaction for them using the database, and then nobody might use it for a few days. So that's a bit different to other use cases where it's a bit more intensively used by more users. So we looked at Aura, which is the cloud offering, but for that use case, it's not the best fit, right? It's more designed for concurrent users and things like that, whereas we don't have that many users. So we looked at starting Community Edition on Compute Engine, and that was okay. So we installed Neo4j on a Compute Engine VM, and then used Google Storage. And then what we can do on Google Storage is download all those data files, build the graph using the commands I've just talked about, import it. Then you have quite a nice provenance there, because you have all the data files and the complete graph. So you know what data made what data, and then you can interact with that data. So what does it look like today? So there's around 3.6 billion relationships between genes, transcripts, variants, and there's also things called EQTLs. And there's also data from different sources that map between genes. So for a given mouse gene, what are its homologs, its orthologs, etc. So what are the links between it and another species? Primarily we're interested in human, of course. So what are those links? And those are actually scientifically arguable. So what I've been doing is importing them with different tags, so that you can actually traverse the graph and then ignore the bits, the kind of orthologs between genes that you might not agree with as a researcher. So and we're still importing some other sources on that for links between genes. And this week we've just been adding links between variants and mouse genes by parsing some of the data that our institution produces. So yeah, all in all, I guess it's around the scale of around 1 billion nodes. So it's, as I say, it's not unmanageable. Just going on to a few examples. So what actually, if you're a researcher, what actually would it look like using this graph? So it wouldn't particularly look like a front end. You could. It is available to people to bring up the excellent Neo4j GUI and try out different maps through these genes. But of course, the graph is relatively simple, as I just showed on the previous slide. It's not a deep graph, right? These links, these EQTL links, there might be scores maybe for between one gene and its variants, and so you're not actually traversing it into a deep graph. But what you're doing is, as a researcher, you're firing off lots of variants at this graph and trying to build up what those individual relationships would be. So you might have a gene set, a set of known variants that are associated with disease, and then you want to look at that through the known links in another species. Okay, so you basically, you might want to go from a mouse into a human. So you've got some variants that you think are associated with disease, or you might have indeed a human variant associated with disease, and you want to know what mice to look at and what the variants should be in those mice. So the first example is actually just saying, for two species, give me a table of all the links between them. And as I said, in the kind of cipher there, we could actually pare that down if a researcher only believed in homology as a link between it. It depends on the data source. Is arguable, scientifically arguable. And then this one actually is actually replacing the need to go and get those files and parse them, and it's not really using the graph. It's just saying, for all of Homo sapiens genes, give me the chromosome start and end position, the chromosome and the start and end position of that gene on the genome. So it's actually not using the graph, but it's a way of replacing what would have been writing R or Python to try and get that table, to actually just running a cipher query. So that's pretty cool. And then this final example is one I've talked about already, where we've got a bunch of variants in a CSV file, and for that we want to traverse the graph, and maybe we want to limit it to certain links between genes, and we want to return a table of genes. And these generally, so the interface for us is a curl command that you actually then execute on the database. Quick note on further work. So we still, as I said already, we're still adding the data sources. So that's our next kind of piece of work to finish off. And then we want to integrate the graph with GeneWeaver.org. So GeneWeaver.org already allows you to build gene sets for diseases that or effects that you might be interested in. Well, the next step would be to be able to put that gene set into the graph, and then get a table of the relationships that the graph thinks exist. We can do further analysis based on tissue using APOC, as I mentioned, and we're thinking of deploying this graph externally. So at the moment, it's on, as I said, it's on a Google Cloud deployment, but that's not public. But we could potentially make that available to other researchers around the US or around the world. And maybe at that point, we would need to go back and look at Aura if it's wildly successful, but I think it would have to be very successful for that to be the case. So in conclusion, so Neo4j scales much greater than our requirements. That's fine. The graph approach makes making queries easy for science. The REST API is extremely useful. We use it all the time. If you use indices, which we use a lot to make our queries faster, then you're going to need the disk space. So obviously, it's like the same for any database that you need. If you're going to write that cache, you need to have the data. You need to have the data space for it. So several terabytes in our case is not huge. And we think that this has great potential. Now, I should probably point out that our solution is actually beta. Although Neo4j is a production system, of course, our solution is still beta. So we haven't done scientific level tests to check that all of the data has been ingested and put into the graph correctly. So that's also some further work we need to do. And finally, just to say thanks. Thanks, Robin Ball, Eric Baker, Jason Boubier, Lisa Chesler, Jake Emerson, Howard Hay, Tim Reynolds, and everybody in the IT departments of the two institutions that are collaborating. And thanks a lot to Neo4j, because they helped me on a few support things. And other members of the GeneWeaver team also got me working with things like Maven Central and things. So with that, I'll hand back to Sanyam.